Uh, Good afternoon and good evening, everybody. I think it's good afternoon for Kerstin and our colleagues from Cambridge, but in Sri Lanka it is evening. It's 6:30 p.m. So I think people are still joining. Maybe we can give them give 1 minute. Kerstin, you're muted.
While people are still joining, can I just point out that my screen sharing is still disabled. So somebody needs to do something so that I will be able to do that.
So it says that you're a host now. I think you've been made a host right now.
Yes, now that should be possible.
Okay, great.
Let me just quickly try just to check that
please.
it's working.
Yes, please.
Yeah, okay, and then I would have to go to here to check. Yes, okay. So I'll unshare again just so that during the introduction we can see everybody.
Yes. So we'll just give because people are still joining. We'll give people just wait for a minute. So we did have quite a large number of registrants to this seminar series, which is quite nice to know. Okay, I think we can start now. I'll just start with the introductions. So thank you everybody for joining us today. This is the first seminar series of single cell sequencing and the Human Cell Atlas. And we are very grateful for our colleagues from Cambridge, Wellcome Genomics Center, working on the Human Atlas, Cell Atlas, for initiating this and collaborating and actually getting this done. So we have an excellent line of speakers during this seminar series who will be speaking on different aspects. I think the program of the 4 days was shared with everybody. And we are very much looking forward to these exciting 4 seminars on learning more about the Human Cell Atlas and single cell sequencing. And and actually speaking, the person who did put everything together is Dr. Diniti Suravira from Teichmann Lab at Cambridge. So thank you, Diniti and her team for making this happen and putting this together. And thank you for having this beautiful program and lining up such a wonderful speakers for this seminar series. So I think I'll start with, so before I introduce the first speaker, so this is Diniti. So would you want to say something, Diniti, before I go ahead and yeah?
So thank you so much for actually giving us support in organizing this seminar series as well. And yeah, very much looking forward to the next 4 weeks and today's talk as well. Thank you so much.
Thank you. So also, we would like to bring your attention to this Human Cell Atlas meeting happening in Asia in 2023, which is in Kolkata, India. So because I can't share this, so because I can't share the screen, I think maybe if Dinit, if you can put it in the chat, all the details in the chat. So I think it's easier for people to then click and follow all the information if people want to register for this meeting happening in Kolkata in India in November this year. So that would be a really good meeting to go to. So without wasting any more time, and because we do have quite a large number of people already, I'll start with introducing our speaker for today. So thank you so much, Kerstin, for joining us and being our speaker for today and kicking off this seminar series. So Kerstin Meyer is a principal staff scientist in cellular genetics, working with the Teichmann Lab. She leads the Human Atlas project, characterizing the cellular landscape of human lung and the respiratory system in development and over the lifespan and in diseases such as asthma and COVID-19. She started her career in immunology, obtaining her PhD or DPhil from the MRC Laboratory of Molecular Biology, University of Cambridge, where she studied the regulation of immunoglobulin gene expression. She continued to work in this field for a number of years before moving to the CRU UK Cambridge Institute, investing in genetic risk for breast and lung cancer. She worked in close collaboration with clinicians and computational biologists, using transcriptional networks to understand the combined effect of inherited risk variants for cancer. She has been fascinated by the way gene regulation networks determine cell types and cell states, and hope that Human Cell Atlas will further understand how cell-based decisions are made and potentially be modified in therapeutic applications. She enjoys working in multidisciplinary teams, bringing together expertise from biology, medicine, computing, and technology development to tackle big problems in modern medicine, which the Human Cell Atlas has the potential to address. So thank you so much, Kerstin, again for joining us. And I hand over to Kerstin to talk about and deliver the first talk of the seminar series. Thank you, Kerstin.
Right, so let me share screen. So before I start, can I just double check that everybody is seeing that screen all right?
Yes, we can see.
Yes, we can see.
Great, fantastic. So first of all, a big thank you to Denithi for organizing the series and inviting me to speak, and to Neelika for that very kind introduction. So today I'm going to give you an overview of the Human Cell Atlas. And as part of that talk, I will first of all explain a little bit why we are so excited about single-cell genomics and why we believe it's such a new frontier in biology. I'll then describe a little bit the Human Cell Atlas as an organization. So how did it come about, and how far has it actually got with sequencing human cells? And then finally, I'll finish on some of the future challenges. But kicking off then with single-cell genomics. I presume all the audience are aware of the central dogma of biology, which is really that DNA encodes the information, which is then transcribed into mRNA, that is translated into protein. And that's really how we have that flow of information from the genetics into the functional side that actually forms the cells. And of course, in the body, there's a whole universe of different cells with an enormous range of diversity. And actually, it's been estimated that there are as many as 37 trillion cells. So really almost, now I wouldn't say infinite diversity, but certainly very large diversity. But amazingly, all of that diversity is, of course, generated through cells that are all encoded by only one genome. For the vast majority of cell types in the human body, the genome is exactly the same, no matter what the cell type is. And of course, that is achieved by accessing different genes. So all the information is present in all the different cell types, but they become different cell types by activating different genes. So this is shown as a little cartoon with 4 different genes that show different expression patterns and therefore have different phenotypes and different form and function. And so why do we want to study this at the single cell level? And the answer is really because that is where the gene regulation takes place. And I've got another cartoon here to sort of explain this a little bit further. Just assume for a moment that this fruit bowl represents an organ. And what we as a biological community have done for many years is really analyze organs through bulk RNA sequencing and transcriptomics. But that's really the equivalent of making a fruit smoothie of this fruit bowl. And you can then see that maybe one smoothie has more kiwi, the other one has more strawberry, and that already has been very, very informative. But I think it's immediately obvious that if we were able to take the fruit from this fruit bowl and sort it out into the different fruit, i.e. into the different cell types, and analyze each of those separately, that you get enormously more information. And of course, it then is really possible to understand how in each single cell, a gene is activated and how that is converted into the phenotypic diversity that we observe. So the Human Cell Atlas really came about because people have long been interested in this question, but they didn't really have the technology to address this. But over the last decade or so, there's really been a revolution in the available technology, and that means that we can now do single-cell RNA gene expression at scale at a reasonable cost, and we can use spatial gene expression technology to map that gene expression back into the tissue context. And of course, both of those are only possible because of large advances in computational analysis. And all of those three aspects together have allowed us to build a human cell atlas. But digging into these technologies just a little bit more, so the single-cell genomics is really where we take a sample from any organ and then dissociate the cells and sequence each of those cells individually. That is then computationally analyzed, and usually the data is displayed in these t-SNE or UMAP plots where similar cells cluster together, and therefore these different clusters represent different cell types of the body. And then secondly, we have spatial genomics, where again, we take a section of an organ, but this time we don't dissociate it, we don't destroy it, but rather it gets placed on a glass slide. And within that, and on that glass slide, there is a barcoding system that allows us to map the gene expression in each of those little voxels on the slide. There's different technology around, but that's a sort of very broad brush overview. And of course, then we can use the single cell data and map that back into each of these voxels, and we can get an estimate of where the different cell types are expressed, and for example, how different cell types interact with one another and might influence each other. And of course, all of this is only possible because over the last decade, along with the technology development, there has been a huge leaps and bounds in terms of how we can analyze this data. And I won't go into that in detail. I'll just show in the next slide a, hang on, for some reason, it is not there. In this next slide, how you can utilize that data. So here's an example of single cells that have been mapped and that are now being mapped back, their characteristic into the human, into the mouse brain. And again, here we see the t-SNE information is converted into characteristics, and then you see it in space. And I think there's a third example of a human lymph node where you can see the different cell types and how they then sit in the tissue section. So all of this together has really allowed us to take a new view of the human body and ask some really fundamental questions. And those are, what determines cell identity, and how do cells assemble to make tissues? And so those two questions are really what has driven the HCA, and I'll now talk a little bit about the organization of the HCA. So in 2016, Sarah Teichmann from the Wellcome Sanger Institute and Aviv Regev from the Broad Institute got together with a group of other scientists who said, you know what, now the time is right. We have the technology to start doing single cell biology at scale, and we should do that in collaboration with people from all over the world. And therefore, in 2016, they launched the Human Cell Atlas. And as a mission statement, they said they would like to create a comprehensive reference maps of all types and properties of all human cells, the fundamental units of life, as a basis for understanding, diagnosing, monitoring, and treating health and disease. So in other words, we really want to build a map, an atlas of all the human cell types. And by building this map, we will really have much better insights into how organs function, and therefore also how we can understand disease. And immediately it was clear that this kind of initiative would have many, many applications. So for example, if we really understand how cells talk to each other and how their regulatory networks work, it'll be easier to understand the disease mechanisms. By understanding where drug targets are expressed, we can better understand drug toxicities, and we can also use that information to discover new drugs. Similarly, over the years, people have studied the genetic basis for disease, and that was often analyzed at the organ level. But by having cell types and cell type specific gene expression data, we're now able to look at the cellular effects of genetic diseases. And another very important aspect I want to highlight is that by being really able to drill into the heterogeneity of cell types, we often also see precursor cells, immature cells, and then finally mature cells within a single organ. And by understanding this differentiation trajectory, we will hopefully be able to translate that into regenerative biology and therapies. So those are the kind of applications that were envisaged. And this is a timeline of how the HCA got going to aim for those applications. So in 2016, there was a kickoff meeting. And then already by 2018, there were over 1,000 members from 53 different countries. The HCA then developed various different working groups. For example, there's a human developmental cell atlas, and there's also an organoid atlas and many others. In addition, it also started setting up a number of different working groups, and all of these have been very instrumental in really bringing the community together. And then, of course, in 2020, the COVID-19 pandemic hit. And many of us working on the Human Cell Atlas, especially those working in the respiratory system, got very much involved into then studying COVID. And I'll come back to an example of that later. Then by 2021, we already had over 50 million cells analyzed and members of over 2,000 members from 79 countries. And so to illustrate that a little bit more, here's just a map of the distribution of some of the institutes and members that are part of the Human Cell Atlas. And particularly for this community, I also wanted to highlight the regional networks that Neelika had also mentioned briefly in the introduction. So in 2020-21, there was an HCA Latin America established. There is an HCA Africa, but actually the HCA Asia has already been in existence since 2018 and 19, has many, many members, and hopefully some of you in Sri Lanka are actually already part of this. And at this point, I think it's also worth saying that the HCA is really a bottom-up organization. In other words, anybody can join. If you feel you have an interest in single cell biology, if you're able to generate some data in this space, you can register on the HCA website and you can make contact with the biological network that you feel closest to. We also have a equity working group, and as the name says, this group is really involved in promoting equity. In other words, we really want to engage with stakeholders across the globe, and we want to make sure that there's representation of samples and scientists that really represent the global world in which we live. Of course, with this effort having started in Europe and America, the majority of samples and scientists come, especially the samples are of European descent, but there are really big efforts now to expand that and get better global representation. We also want to empower global participation via training and workshops. And of course, this lecture series is already part of that effort. So where have we actually got to at this point in time? Have we actually already got a sort of guide and a blueprint of the human body? And we may not be there yet, but actually the HCA community to date has sequenced approximately 120 million individual cells, either it's a single cell or single nucleus level. And that is enormous progress, considering that it's really only been going for 6 or 7 years or so. Now, again, in a bottom-up effort, the HCA has actually organized itself into different biological networks. And in particular, I can highlight a few here. There's, for example, the kidney network, there's the lung and respiratory network, the skin, developmental, and so forth. And again, the idea is that by bringing together these networks, people reaching out to the HCA will have a point of contact where they can find other like-minded people. Oops, and there's something's gone a little bit wrong on my screen. I'm not sure whether you're all seeing this too, but I'm just getting rid of something on my screen. So we are back to my talk. Apologies about that. And so now I would just like to give you two little examples of studies where the HCA has, in my opinion, really contributed to an understanding, and studies that sort of highlight the impact the Human Cell Atlas can have. The first one comes from the human, comes from COVID. As I already mentioned, the Human Cell Atlas was not very old when the COVID pandemic hit. And of course, we knew relatively early on through its homology to the earlier SARS outbreak that the SARS-CoV-2 virus binds to the ACE2 receptor. And by having a Human Cell Atlas, which charts gene expression in every single cell type, we were immediately able to go back to this atlas and ask, where in the body is the ACE2 receptor expressed? And of course, what we found was that it was expressed in the airways and the lungs, as expected for a respiratory disease. But we also saw that there was high receptor expression in the oral and nasal cavity, as well as in the cornea and the conjunctiva. And by being able to map these viral entry factors, that then had impact on public policy and on understanding the routes of transmission of this virus. It was also really interesting to see, for example, that in the gut, enterocytes also express ACE2. And of course, that is consistent, for example, with the fact that people suffering from COVID-19 often have diarrheal symptoms. There was some expression in some cells of the heart, which again goes with the observation that people with heart comorbidities were more severely affected by COVID. So I think that's a very nice example of the utility of a Human Cell Atlas. The second example I want to show has to do with developmental biology. So this is a human developing lower limb in post-conception week 5. And here we've identified different cell types that are highlighted by these different colors. And this time, the presentation is not just in 3D, seeing as it turns around, but we're really going to 4D because, of course, we're looking over developmental time. So the first images were from post-conception week 5, and now we're moving to post-conception week 6, where you're beginning to see the digits forming and the bone structure forming and so forth. And we are able to mark these different cell types and are really able to zoom in and understand how cells sit next to each other and influence each other's development. So that's the only science vignettes I'm presenting, but of course, there are other lectures in this series where you'll hear more science. So I now want to come back to the overview and some of the challenges that we face in the coming years. So the first one is that we started out using primarily single-cell RNA sequencing, but of course there are many, many different other ways in which we can profile cells. So we might just want to look at the transcriptome, but we could also look at the open chromatin, so doing single-cell attack-seq. We might want to really know about the proteins because, of course, RNA gets translated into a protein. And then, of course, we don't just want to have those different modalities, but we also would like to have that information over the whole of the human lifespan. We would then like to integrate all this multimodal data with the spatial data that we're generating, and then taking that spatial idea one step further. Ideally, we are aiming to include this with high-resolution imaging, for example, high phase contrast computed tomography. And of course, CT scans are being used as part of clinical work. So that would then really allow us to take this molecular profiling of cells into a clinical setting. And then lastly, of course, all of this involves a lot of computation, and data sharing is absolutely fundamental for the success of the project. And again, this is something that needs further support. But taking all of these together, our vision of where we would like to go is really summarized in this figure here, where we have a molecular description of the multi-scale human, so that we're really understanding processes all the way from the nanometer scale, where proteins interact, to organelles, and then larger structure all the way to organs, and of course the whole human body. So it's going to be a long journey, but a very exciting one, I think. Coming back to the data sharing, one way of involving other researchers from other communities is to widely share the data through data portals. And in particular, we're aiming to generate browser-based portals that can be accessed from anywhere, and also that can be used by non-computational experts. So maybe clinicians or educators who should be able to access that data and use it for different purposes. We want to use FAIR principle in the data sharing, and that stands for findable, accessible, interoperable, and reusable data. And this reusability will really then also form the basis for collaboration, so that we make the data freely available to anybody who's interested. We would like to extend the global engagement, as I'd already said, and this should really happen in the spirit of a true knowledge exchange, so that other researchers can take the foundational knowledge, apply it to local conditions, and ask local questions, so that we're really talking about co-creation of new knowledge rather than knowledge delivery, if you like. We will, of course, want to apply the Human Cell Atlas to disease. And although the Human Cell Atlas community at the moment is very strongly involved in building HCA references, there's already a lot of disease data out there. And so that one of the next steps certainly is to include those disease data set in the reference atlases. We would also like to build in vitro models, so that some of the insights that arise from the Human Cell Atlas can be tested experimentally. And then lastly, a program that would involve a lot of scaling is to really represent the breadth of human genetics in the Cell Atlas. We're not there yet, but the Human Cell Atlas is now funding different projects to really look at the influence of ancestry and ethnicity on single cell behavior. And for example, there's the Asian Immune Diversity Atlas, which is a very exciting project. So in conclusion then, we are building a human reference atlas that is beginning to take shape. Our future aim is to include further genetic variation. We would like to include disease data and in vitro models, and all of that, of course, requires a lot of computational power and innovation to really make the most of the Human Cell Atlas. And with that, I'd like to stop and thank you very much for your attention, and I'd be happy to take any questions.
So thank you, Kerstin. That was just an amazing talk, and with just amazing potential of, where all of us could be heading. And so before taking questions, just about your talk, the beauty of it, I think, is, the global involvement. And unlike what has happened in the past, where, people just collect the data and, keep it yourselves, here you are actually engaging with the global community so that they can take things forward and have their own data sets to work on problems they have, which might be very different, in different parts of the world. And but just, I know you are already, because they did say that you are working with certain countries in Asia, because if you look at a lot of data, there is the data available when it comes to single cell sequencing or genomics or whatever is mostly in the global north. But populations, if you look at, African populations, Asian populations, we know that when it comes to a lot of diseases, how actually they develop, certain disease phenotypes is different, even like metabolic disease, and even simple things like how they might respond to drugs is different. So I just want to know whether your I know you showed the countries you're actually looking at, but I think that's the really beauty of things, that you want to look at all these different populations. Yeah.
Yes, so maybe if I can just comment on that. Yeah, so I couldn't agree more. I think it's incredibly important to look across ancestries. And prior to joining the Human Cell Atlas, I actually was working on genome-wide association studies. And of course, they have shown very clearly that there's different disease susceptibility in different populations. And that surely is reflected in the way individual cells are behaving, because like I discussed, that is where the genotype is translated into a phenotype. And yes, it is true that at the moment, the vast majority of samples in the Human Cell Atlas are from Europeans, because of course, any lab setting up experiments will use samples from the local hospital. And for example, a lot of the samples that we've analyzed in our lab come from organ donors in our local Cambridgeshire hospital, and they are primarily white. But I think as we're going forward, the technology is changing really, really quickly, and it's becoming easier and easier to preserve samples. And also technology, different types of sequencing are becoming available that are not quite so expensive as some of the early iterations. And I think both of those aspects will really help to extend the reach of single cell sequencing. And I mean, there are funding bodies who've really recognized this. And for example, the Chan Zuckerberg Initiative, who's been a great supporter of the Human Cell Atlas, have specifically had a grant call to improve the diversity of the samples that we have. There are also, over the years, we've had some collaborations with Thailand doing single cell sequencing on dengue fever. So, there are individual projects, but I think really ideally, your community in Sri Lanka should really think about what are the questions that are really burning here, and how can we set up experiments so that we can, make the most, given our local constraints, but make the most of the technology and the insights we can get through this.
Yes, I can think quite a few of the burning areas, like burning questions, and how, limitless, endless things we can do with such technology, especially in our populations, and also the unique disease phenotypes that we see in our populations, like let's say South Asian populations compared to, let's say, Caucasian, or even, East Asian or the African populations. I think, you do see very different disease phenotypes, susceptibility to disease in South Asian populations compared to others. And of course, resistance to disease as well. I mean, it's both ways, not just susceptibility, but, resistance and milder disease in certain instances with certain infections. So I'm not only talking about infections, but things like metabolic disease and so on. So that's really great that, if you can, for us in Sri Lanka, it's not just about accessing the technology right now, it's about finding funds to set about, using the technology and establishing things here. But I'll stop there, and I'll stop talking and invite questions because we have quite audience here. So, so anybody want to put their hands up and ask a question, or anybody wants to put the questions in the chat?
And this is really meant as an engagement. So, ask anything you like or any comments you have. It doesn't, any contribution would be fantastic.
Yes, so because I don't want to, dominate the conversation here, but anybody who is feeling shy to put, switch on their camera can, put questions in the chat. Well,
And of course, this was also, it was very much the sort of big picture view, and there will be talks that give more sort of detailed descriptions on some of the projects that have been happening in the Teichmann Lab, and it might be easier to, you know, have specific questions for that.
Yeah, but I think, you know, with general questions also, I mean, there's such a lot of, you know, potential, not just potential, I mean, limitless potential, I would say. Anybody has any question or comment? Asanda, yes, please, Asanda.
Yeah, thank you, madam. Also,
if you can be a little bit louder, please. Sorry.
Yes. So I have a question that I'm much interested in the macrophages and the dynamics of macrophages in transcriptomic dynamics in dengue fever. So in that case, this Human Cell Atlas project seems to be a good reference for me to start as going forward, so that is it possible for us to get like the pipeline or the work protocols that the Human Cell Atlas project using to get the and process the data from different I think I'm pretty sure that this is not the single cell sequencing is not done with single technology like Illumina or Nanopo. It may be combined technologies. So is it possible for us to get the the perfect easy, reliable, cost-effective working pipeline to process go forward with our researchers on the single cell sequence? Thank you.
So I think your question touches on two things. So even though I've presented the Human Cell Atlas like as an organization, but of course, each individual study is driven by a particular lab, and it's really the responsibility of that lab to make sure that all the data that is being generated is being shared and that the technologies are being shared. But for the majority of Human Cell Atlas papers, we put all the protocols that are being used on protocols.io. And so if you go to the humancellatlas.org website, there is a link to protocols.io, and you'll find a lot of the sort of the cell type dissociation and preparation protocols there. And similarly, all the computational tools should be in various GitHub repositories that will differ for the different papers. But of course, all the actual data that's being generated is provided through a number of different portals. And actually, I had a last slide that I didn't share, so maybe I should do that and go right to my very last slide and see whether you can. So on the humancellatlas.org, there are a number of different portals where you can already download data that is published. So I would recommend that you go to some of these portals first and see if you can already find some of the dengue data there. And if you can't find it, then do send me an email. I can see whether I can connect you with the people doing that work. I won't keep the slide up because I'll stop sharing, but all these different portals and the links were just taken all from this overarching humancellatlas.org webpage. So, it should be relatively easy to access. So let me just stop sharing again. I'm not quite sure what my share button has gone. Right, so
yes, so we've got another question in the chat. Does the Human Atlas Cell Atlas support in cytospace or any visualization tools, those networks? Thank you. Yeah, amazing and informative talk. Yeah.
So we don't really directly link to cytospace, but there are many different tools out there for data analysis, and they're really great online tutorials for starting out with starting to look at network biology. I think, I mean, there's obviously lots of different types of networks and lots of different visualization tools. So it's quite an active, it's a very large area that is covered by your question. But I'm just trying to think where, I mean, a lot of the computational tools from our lab are on the Teichmann GitHub repository. Another group that's done a lot with networks is the TICE group in Munich, or maybe Dana Pear's group at the Broad. So I would go to those websites and see what you can find there.
Any more questions or comments? Okay, yeah, so what are the common challenges in this method? I think that's a very, very broad question.
Yeah. So the first challenge in generating single cell data is to obtain good tissue. You know, it's, you know, in that sense, single cell biology is no different to any other biology. So if you're wanting to get good human data, you need to make sure that the samples that there's a very, very short warm ischemic interval. So when the sample is taken in the clinic, for example, let's say there's cancer surgery, you are able to take normal neighboring tissue, that tissue is then stored at 4 degrees very quickly in preservation solution, or is processed very, very quickly. Luckily, a lot of those challenges are becoming are easier to overcome now, because people have shown that you can fix tissues very effectively and still get good single cell or single nuclear data out. And I think so that's become a lot better. You then obviously have to go through the single cell sequencing itself, but there are a lot of different companies now who offer products, and they're really quite well supported. So I think in that space, your biggest challenge is actually having enough funding to do the work. And then at the end of the process, the challenge is also having the computational know-how to really interpret the data. But once again, the tools are really being made available to make that easier. So, for example, manual cell type annotations used to take an enormous amount of time, but there are now automated tools that have made that much easier. Things like Cell Typist from our lab, or Azimuth from the Satija lab. And hang on a second, I need to plug myself in, because otherwise I might just be lose you all. So that's better. So I would say there are challenges all the way from the experimental through to the computational. But as I said, I do really think that more and more tools are being made available to support people who want to start this up.
So thank you so much for that, Kerstin. We've gone like just about 3 or 4 minutes over our time. I think has anybody else got any questions for this evening or afternoon, as at least for Kerstin? Yes, since there are no questions, I would like to thank Kerstin for this amazing talk and like a really good beginning for this seminar series. And thank you so much, Diniti, and the rest of Diniti's team for organizing this. We are very privileged to have Kerstin speaking to us, and we are looking forward to the next seminar. So thank you so much, everybody. Have a pleasant evening.
Thank you for having me. Bye, everyone.
Bye.
Thank you.
Thank you.